I hope you said there's an excess of homozygotes. In a non-evolving population, we expect 56.25% big A and 6.25% little a, and we have 60% and 10% respectively, both numbers bigger than the corresponding g-hats. In the non-evolving population, we expect 37.5 to be heterozygous, and we have only 30% in our actual population. This numerical example is a case of excess of homozygotes. In an alternative example, I might have told you that the population consisted of 50 red-flowered plants and 50 pink-flowered plants, and no white-flowered plants. Work out the allele frequencies for this population, and then work out the Hardy-Weinberg genotype frequencies for the theoretical non-evolving population. Okay, for allele frequencies, you should have come up with P equal to 0.75 and Q equal to 0.25, the same as what we had in the first example. And for the Hardy-Weinberg values, they're the same as the first example as well because we're using the same allele frequencies to calculate Hardy-Weinberg. In this case, however, there are fewer homozygotes in our actual population, 50% instead of the g-hat of 56.25% for big A, and 0% instead of 6.25% for little a. And the heterozygotes are too many relative to Hardy-Weinberg, 50% rather than 37.5%. Both this and the first example show departures from Hardy-Weinberg, but in the second example, there's an excess of heterozygotes. It's also possible for the actual population to match up very closely with Hardy-Weinberg expectations. If I had told you there were 56 red flowers, 38 pink flowers, and 6 white flowers, your numbers would be really close to matching to those of the theoretical non-evolving population, as close as you could get, given that the numbers of plants had to be reported in whole numbers. The departures of negative 0.0025, positive 0.005, and negative 0.0025 would not require explanation beyond the fact that in a sample of 100 plants, we really couldn't match the theoretical calculations exactly without increasing the resolution to quarter plants. You couldn't actually have 56 1/4 red-flowered plants, could you? But let's get back to the core challenge of this exercise, which is all about the limits of what can be inferred from the information provided. Let's go back to the 60 red, 30 pink, and 10 white numbers that I gave you originally. What exactly can you infer from this? As I said before, you can't tell from a single snapshot in time whether the population is evolving toward becoming more red or more white. That would require at the very least information from 2 times, a before and an after. But with Hardy-Weinberg, we can actually assess whether the population matches the description of a non-evolving population. And if it doesn't match, we can infer that either evolution or non-random mating must be the cause. Null models. This inference exercise using the Hardy-Weinberg law is a great example of how a null model, or null hypothesis, is used in science. In the prototypical scientific study, we start with a model that asserts something about the natural world. This is our hypothesis, by which I mean our explanation of what's going on. Then we ask whether or not the observations we make are consistent with it. Now, this is going to sound weird, but in order to demonstrate support for our hypothesis, what we actually must do is to demonstrate that our observations are not consistent with what we would expect if our model is wrong. The hypothesis stating, whatever Jeff said about the natural world, that's not true at all. This is the null model. Furthermore, this null model must make a specific prediction that's different from what my model predicts. When the results come in, the first thing I need to show is a good reason for rejecting the null model. Only then can I even entertain the possibility that my model has support. If I can't reject the null model based on my results, this would not be considered as valid support for my model. Take our plant growth experiment. We hypothesized that adding fertilizer will facilitate plant growth by providing nutrients that might otherwise be limiting. And in our experiment, we grew plants under similar conditions but manipulating the amount of added nutrients, a high nutrient environment and a low nutrient environment. Up to here, everything is familiar to you, right? Now it's time to dissect our experimental procedure more formally. You might think that it's our objective to show that high nutrients result in greater plant growth. That seems reasonable enough given what we're doing in our investigation, but this is not entirely accurate. For one, no matter what our results are, we can never really show our hypothesis to be true to the exclusion of all other explanations. That much is something we just have to accept. There is always another factor that we're not considering that might be the actual cause of our results, and this will be true no matter how dutifully we carry out our experimental controls. What we identified as our hypothesis, that adding fertilizer helps plant growth, is actually an alternative to the null hypothesis, which states that adding fertilizer has no effect on plant growth. H0 is a symbol for the null hypothesis. Moreover, this null hypothesis makes a specific prediction about our experimental results. There should be no difference between the high and the low nutrient plants. Our hypothesis is actually called the alternative hypothesis, HA, and this becomes a possible explanation if we are able to reject the null hypothesis based on our experimental results. We designed our experiment in a way that will allow us to do exactly this: reject the null hypothesis if certain results were obtained. If our data were to show a statistically significant difference between the high nutrient and low nutrient plants, we would have a valid reason to reject the null hypothesis. It predicted something that was demonstrated not to actually happen. If, however, there were no statistically significant differences between the two treatments, then we would not be justified in rejecting the null hypothesis. And this means that our alternative hypothesis is unnecessary to account for the results we got. It would also mean that we have no more justification for thinking that our hypothesis is true than we did before doing the experiment at all. I know this probably sounds completely weird to you if you haven't heard it before, but it's true. In scientific experimentation, we never really set out to prove our hypothesis. The absolute most we can do is to reject a null hypothesis based on the absence of the mechanism we propose in our alternative hypothesis. Mathematicians, they can prove things by showing that y is a logical consequence of x. If x is true, then by necessity y must also be true, QED. In this regard, experimental science is actually a very different enterprise from mathematics. We get hardly any opportunity to carry out anything remotely resembling proof. Getting back to our evolutionary discussion now, the Hardy-Weinberg law gives us a specific set of predictions: that the genotype frequencies of big A is p squared, the genotype frequency of big A is 2pq, and the genotype frequency of little a is q squared, under conditions of no microevolutionary forces. If we have a non-evolving random mating population, we will have Hardy-Weinberg equilibrium genotype frequencies. So if we don't have Hardy-Weinberg, then what? Right. Given that the 4 microevolutionary forces plus non-random mating are the only possible causes for departure from Hardy-Weinberg, then this means that if we have numbers like these with an excess of homozygotes, or numbers like these with an excess of heterozygotes, this must require that one or more of the microevolutionary forces, or non-random mating is operating in our population. In other words, the population is evolving. So even without looking at changes over time, because we have only a single snapshot of the genotype frequencies at just one time, we still have an opportunity to reject a null hypothesis stating that our population is non-evolving and random mating. This is how we use Hardy-Weinberg as a null model. It provides a set of expectations for a non-evolving population. If we find that our population conforms to Hardy-Weinberg, we have little choice but to accept the null hypothesis that, at least from the limited information available, there is no evidence that the population is evolving. No evidence for non-random mating. This would be the only reasonable inference. However, if we find that the genotype frequencies realized in our population depart significantly from Hardy-Weinberg, we may be justified in rejecting the null hypothesis. And this means that our population is not a non-evolving population, and therefore it must be evolving. At that point, it may be reasonable to propose an explanation for the observed departure, maybe one or more of the evolutionary forces, and maybe non-random mating. As we said before, there are two ways in which a population can deviate from Hardy-Weinberg. You can either have an excess of homozygotes, or you can have an excess of heterozygotes. If you find a departure from Hardy-Weinberg, the explanation that you propose to explain it must be consistent with the kind of departure that you're seeing. For example, if you find that there's an excess of homozygotes in the population, as we did with our original Snapdragon example, then a natural selection-based explanation would need to account for a fitness advantage for both big A and little a as compared with the heterozygote. Red flowers and white flowered plants have higher fitness. Pinks have lower fitness. This is the only kind of natural selection that would be consistent with the excess of homozygotes we have for our single snapshot in time population. You should also try to envision some real-world mechanism that would account for these fitness differences. Maybe pink flowered plants are more visible to deer who then seek out pink flowered plants to eat, or something like that. This would give them lower fitness. Put together a story that would be consistent with natural selection, resulting in the deviation from Hardy-Weinberg that we're observing. Conversely, if there had been an excess of heterozygotes, then they would be the ones with a higher fitness. Maybe rabbits avoid eating pink flowered plants because the pink flowers give them indigestion. Now, the elephant that's been in the room for a while now is non-random mating. And yes, it is a potential cause for a departure from Hardy-Weinberg. You need to be really clear about what non-random mating is and what it isn't. We're talking here about a specific phenomenon that is distinct from natural selection. For non-random mating, you should presume that the fitnesses are exactly the same for everybody in the population. That is, no natural selection. The randomness in random mating actually refers to the likelihood of ending up in any of the possible matchups. If I am a snapdragon plant in a population that is 60% red flowered, 30% pink, and 10% white flowered, random mating means that, irrespective of my own flower color, my likelihood of pairing up with a red plant is 60%. My likelihood of finding a pink flowered plant as my soulmate is 30% and 10% for white. Under random mating, this would be true for everybody in the population. Everybody has exactly the same set of likelihoods of mating with the three genotypes corresponding to the frequencies in the population. Non-random mating takes one of two possible forms. You can have positive correlation between mates, in which individuals are more likely to select a mate of their same genotype. If I'm a red flowered plant, my likelihood of mating with another red flowered plant is higher than the 60% frequency of red flowers, and my likelihood of mating with a pink flowered plant or a white flowered plant is lower than their respective frequencies. Or you can have negative correlation, in which you're less likely to mate with another individual with your same genotype than what would be expected under random mating. One example of this is negative assortative mating, or opposites attract. If white flowered plants were somehow more attractive to and attracted by red flowered plants, you'd have a greater abundance of matings with one parent big A and the other little a. This cross of big A with little a results in all heterozygous offspring, right? Negative assortment means that you'd also have fewer matings within the same homozygous type. Not as many big A crossed with big A, and not as many little a crossed with little a. Again, this is relative to the expectations under random mating. Now these are the very matings that give you all homozygous offspring, and consequently, negative assortment results in an excess of the matings that give you all heterozygotes and a shortage of the matings that give you all homozygous offspring. And so you find 1. A departure from Hardy-Weinberg, and 2. An excess of heterozygotes. Negative assortment could be a cause for this. Now, positive correlation between mates has a handful of possible causal mechanisms. One of them is positive assortative mating, in which organisms are narcissistic or mildly racist or something like that, and prefer to mate with other individuals that look like themselves. Another possible cause is inbreeding. Maybe there's a tendency to mate with related individuals. Your blood relatives are more likely to carry your same genes as compared with people picked at random from the population, and so the result is a higher degree of homozygosity than what would occur under random mating. When the blood relations are close, as in the case of within the extended family, you would call it inbreeding. But inbreeding doesn't need to be so terribly blatant and yucky. Plain old geographical distance results in a mild form of inbreeding. If you're a plant growing in Oceanside, but your population stretches across all of California, are you really going to receive pollen from plants that are a representative sample of the genetics across the whole state? Of course not. The plants whose pollen sticks to your stigmata are mostly from your area, and in your area, there's a greater abundance of your genes kicking around. No matter what the mechanism, positive correlation results in an excess of homozygotes. If you want a more mathematical demonstration of how this occurs, as well as complete mathematical proof, yes, a real proof, that the absence of evolutionary forces coupled with random mating leads to a non-evolving population at Hardy-Weinberg equilibrium genotype frequencies, I invite you to view a couple of optional videos, the links to which I've put in Canvas. In the meantime, I want you to be prepared to work out a problem based on inference about a population's evolution using the genotypic composition that I give you of the population from a single snapshot in time. This will be one of the challenges in the first exam. This might seem like an excessively difficult problem, and I would feel terribly guilty about this if not for the fact that I'm giving you advance warning, as well as plenty of opportunity to practice and prepare yourselves. In Canvas, I have posted 3 sample problems that you should complete on your own for practice. During the lab activity on phylogeny reconstructions, I'm also going to be calling on 3 volunteers to work out solutions for these 3 sample problems live in class. Work out the solutions for yourself early and share your approach as one of the 3 volunteers, and you get the reward of 5 BET extra credit points. In the time I have remaining, I'd like to follow up on something that came up in our little discussion on null models. Why it's appropriate to prefer the null model as compared with the alternative model. Take our plant fertilizer example. If the data came out showing a little higher average plant weight for the high nutrient treatment compared with the low, but the difference is not enough to be statistically distinguishable from the result of no difference, this would be kind of an in-between result, right? There's a difference, but it isn't enough for us to reject the null hypothesis outright based on a statistical analysis of our experimental results. We also can't exclude the possibility that our hypothesis is actually the thing that accounts for the little difference that we observe. But in choosing between the alternative hypothesis HA and the null hypothesis H0, we go with H0. This is basically saying that until you can reject the null hypothesis, you have to prefer it over the alternative hypothesis, the one that you thought would best explain the natural phenomenon that you're investigating. If the data indicate a toss-up, why should the null win every time? Why should it be the default? Parsimony means simpleness in the sense of having the fewest requirements. In this class, we will on occasion resort to parsimony-based argumentation to evaluate the relative likelihoods of truth between different hypotheses that compete as explanations for something. If a phenomenon could be explained by hypothesis A, which requires that 5 independent conditions be met, and alternatively by hypothesis B, which requires only one condition, and it's not something outlandish. The condition has to be a reasonably plausible one. The principle of parsimony says that hypothesis B has a greater chance of being the actual truth because it's more likely that B's one condition be met as compared to the 5 conditions required by A. The principle of parsimony, sometimes called Occam's razor, is actually a big part of science. As I said, we're going to be calling upon this idea frequently when we have to evaluate between alternative explanations for the same thing. Now, parsimony is a guideline, not an infallible rule. Sometimes the right answer is the one that involves more steps. This is because nature is not constrained to take the simplest path, and sometimes it doesn't. You can't count on parsimony to always lead you to the right answer, but with all else being equal, you're wise to put your money on the explanation requiring the fewest moving parts. Null models are inherently more simple than their alternatives. By their nature, they say, nothing interesting is happening here. Remember our working understanding of how science works. In order for you to propose positively that something is happening, you need to first demonstrate that nothing is not happening. This sounds silly the first dozen times you hear it, but eventually you'll realize that this is exactly what a scientist does. The reason why we do things this way is that nothing is a simpler explanation than something. In my view, the folks in the ID creationist camp like Michael Behe and Bill Dembski think that they're using this basic scientific format. That by rejecting the hypothesis that mechanisms based on natural selection are capable of explaining life's complexity, they're making room for the proposal of their preferred alternative, a supernatural explanation. But it's not the same. The difference is enormous. The scientific method involves acceptance or rejection of a null hypothesis. Nothing is happening is one naturalistic explanation. And if the null is rejected, then an alternative can be proposed, and this HA is also a naturalistic explanation, also based on the laws of nature. The ID people mess this up by saying that the absence of a known naturalistic explanation, such as natural selection, opens the door to the proposal of a supernatural explanation, and I'm sorry, but that's just stupid. Another very important name for you to be familiar with is that of Sir Karl Popper. He's the author of this framework of science we've been developing in this lecture. The idea that scientific theories are abstract and can only be tested indirectly, not through direct proofs, but rather through showing that they're not demonstrably false. And this reminds me to point out that the main way you'll be hearing reference to Popper is with the criterion of falsifiability. And this is commonly called the Popperian requirement for something to be considered scientific. There must be a result that would show a theory to be false. Anything lacking this ability to be shown false would be outside the domain of science. The ID creationists fail because they're treating natural selection as a null hypothesis. It's not, of course. And they're also proposing a supernatural explanation as the alternative, which fails because it's not falsifiable. As David Attenborough pointed out in the video, the problem with supernatural explanations is that they're able to explain everything. And by being able to explain everything, in reality, they explain nothing.